Hello everyone, my name is Ashfaq Ahmed and I work in the field of structural biology and bioinformatics. In our last video, I performed a tutorial that how we will analyze the nucleotide data in Google Colab for phylogenetic analysis. Today, I'm going to perform or to teach you how you can generate or how you can perform phylogenetic analysis on protein sequences in Colab. It's a complete tutorial just like before. So let's start. Okay. First of all, you have to come here, uh, Google Colab Research dot google.com GitHub H100. Here you can see, or if you cannot see, or you have some problem in your internet, then I will drop this link in the description section of this video. So take it from there. Then once you click here, the portal is not updated, so that's why I have to rerun it. Okay. Then you will see this kind of window. Click here, come to alignment H100 forward slash alignment. Now here you will see protein analysis, uh, phylogenetic analysis protein sequences, and this one is the previous one. So click here to open this notebook. Now you have to connect the notebook here. This is all about the introduction of this tutorial. Uh, in this tutorial, we will deal with protein sequences and we will upload a sequences. Then we will perform alignment by using a program muscle. Remember, in our last tutorial, I used uh, MAFT, MAFFT, but today I'm going to use muscle. Uh, I'm doing this because I want you to know that you can use muscle, also you can use mouth and so many others if you know little bit of these programmings. Uh, Secondly, muscle perform best on protein sequences and tea coffee perform best on nucleotide. It's from the literature I'm claiming this point. So, okay. Then after alignment, then we will perform phylogenetic analysis or some phylogenetic calculation using IQ tree, and we will explore different evolutionary models and then we will choose the best model. And finally, in this section, I will also teach you that how we can visualize the distance matrix between your sequences. And this is all about me. And if you are already watching this tutorial on YouTube, so this is a link. If you want to cite this work in your research paper, so this is the muscle citation, this is the IQ citation, and this is the model finder citation of IQ. So first of all, we are going to run this cell. It will install muscle. Also, it will install the IQ tree program and it may take almost 30 seconds. So let's see. Okay, 12 seconds. Now you will see cannot state IQ directory the moving error, but ignore it. Uh, you will not face any problem because of this. Okay, let me clear this output now. The second step is upload your FASTA file. Let me show you on the desktop. My files are here in the data folder. Here's the data. This is my file. I will use it. Uh, okay. It contains different sequences, almost, I think, 13 sequences of albumin. This is from Homo sapien, this is from gorilla, this is from Penascus, etc., etc. If you don't know how I got these sequences, so I have already covered the retrieval of sequencing part in the Learn Bioinformatics from the Scratch series. So I would suggest to watch it there. But for your information, I got it from Blast with some sort of filtration. Okay, so first of all, the notebook will treat the sequences in the FASTA format, and you can see I have the text format. So I will use the program BioEdit. It's the simplest program in the field of bioinformatics. You can use it desktop, and then I have a data directory, and the data directory contains protein sequences. These are those 12 sequences. They are not aligned. You can see uh, from this sequence. You can see, and you can also not see gaps. Also, you can see unequal length. So it means the data is not aligned. So first we are going to save it in FASTA. Let's say I will save it in the FASTA, and name will I will give it a name. Let's say uh, throat underscore seq. Okay, and the extension should be this one, FASTA. Save. Okay, let me check. Yes, we have this file now. Okay. Now come back to the notebook, upload FASTA file, run this cell, choose file, then I have data directory, throat underscore sequence FASTA. It will be uploaded. Yes. Now, in this step, we will use muscle to align our dataset, and you can explore here whether your data set is uploaded or not. So this is our dataset throat underscore sequence and dot fast, or its extension of FASTA. Okay. So if you want to review this code, click here, and they will just uh, run this file. But if I run it, it will throw an error. Let's see. Okay, most of you guys just quickly comment on the YouTube channel, although I reply almost all the comments, but still there are certain errors which you can fix it by yourself. It says cannot open content sequence retrieve dot fast. So why it cannot open? If we see here, our the name of our this is the muscle function. It will treat the FASTA file, but if you see, the name of the FASTA file is sequences underscore retrieve dot fast, but we have uploaded protein underscore sequence dot fast. So click here, copy path, and do this. Now you can see content forward slash throat underscore sequence dot fast, and the out means it will generate the aligned file, that is align.fasta. So you can run it again. Now it's done, and you can see align.fasta, a new file came here. If you want to analyze download, and let's open this download in the bioedit. Okay, it will be in the downloaded folder, I hope so. Yes. Now you can see gaps and also gaps here, but they are now perfectly aligned. Okay, remember, if your data is bigger, if I'm not wrong, I have let me confirm again. we have 13 sequences and the length is not very bigger, almost 600 amino acids. So if you have many sequences, so it may not be so quick. It will take some time. So be passionate for that. Okay. Now, this is the step where we are going to run the IQ tree. And the IQ tree, remember, the IQ tree offers you different options. For proteins, it offers you these models: LG, WAG, JTT, DEHOF, VT, MT, ART, and HIVB, HIVW. It's important for you to understand if you are running some general protein sequences and if you are not good in bioinformatics so much, then I would suggest to choose the LG model. Okay. But if you are running on globular protein and you have some kind of knowledge, like these are specifically globular protein, then you can also use WEG, also you can use LG. If you are interested, if you have collected your proteins from different families, just like if you remember PFAM database or EnterProScan database, before they were separated, but now they are merged into a single database, EnterProScan. So if your research is based on some family data, then you have to select JTT model. If you want to use the earliest models generated by Migrate and DAHOF, then you can use this one. But most of the guys, they are dealing with mitochondrial proteins from vertebrates, then it is the specific model in IQ3 is the VT model. Okay, and these are then further specific mitochondrial models. Okay, but if you are using some viral proteins, then I would suggest to use this one. Okay, these are then additional modifiers that how these models will treat your data. This is for gamma distribution, this is proportion of n variable sites and empirical amino acid frequencies and free rate model for heterogeneity. Okay, so today we are going just to cover the tutorial, so I will not go to the classical or the lecture portion of this video. So I would suggest if you are following another research paper for your data, then follow them. But if you are not following and if you want to know the use of these things, I would suggest to read some good. You will find these things in a few research review papers. If you can read, it will solve your problems. Okay, but what will you do if you are new in the field and you don't want to read so much research papers? Then what will you do then? So there is an answer. The IQ tree provides the answer. We call it a model finder. So that's why I also kept the citation of model finder of IQ here. Okay, what the model finder will do? The model finder will just take your data as a input, then it will check all the models on your data with all these additional modifiers, and then based on the some scoring scheme, we call it BIC scoring scheme. Based on that BIC scoring scheme, the IQ tree will decide which model was the best in treating your data. So this is the second option. Okay, so let's run it. Okay, once again, this step throws an error, but if you read the last line, it said files not found. So let me check here. So now here is in this whole script, there is only one functional code, that is this one. Okay, This one is actually the function from here to here is the function for IQ tree to run. Then uh, after this flag, you can see content 0 dot fasta. Okay. It means this is the input file. Remember, your first input file, which we uploaded, that was we made the protein underscore sequence dot fast. This one was uploaded, but remember this file was not aligned, and IQ tree will only consider the aligned part. So we have then generated this aligned FASTA. So we will use this aligned FASTA here. So how to use it? Click here, copy path from here to till the end of this FASTA and click Control V. Now we have content aligned FASTA. Then which model we will use? So as I told you, let's say you are interested in the LG model with gamma distribution. So it will become LG plus G. So you can change, you can write here LG plus G. But if you are not going to use some specific model and you are interested to ask IQ tree, like you treat all the models on my data and then tell me which one is the best. If your data is a bigger one, then let me tell you, it will take some additional time because the program will test all the models. So if you are dealing a bigger data, I would suggest you should know which model you are interested to know. So the program will directly only calculate for that specific model. Okay. Now here we will use the model function finder. Okay, and then this BB means bootstrap. So by default, I kept it 1000 bootstrap values. You can increase or you can decrease. It's up to you. Okay. Now we are going to run this IQ tree. Now it has started running. It started running. So let's wait until it's finished. Then I will tell you uh, what kind of calculation it has generated. It may take almost a minute. I'm not going to pause the session, so just wait. Okay, it's done. Now let's quickly analyze for your further understanding. IQ tree is initialized and it took all your 13 sequences. In my case, 13. In your case, it will be different. Okay. Then this table tells you for different model, you can see LG then with plus I modifier, then LG with plus G4 modifier, then LG with plus I plus G4 modifier. As I told you here about these modifiers. So here, okay. So this step check all the available models, then generate it, we call it BIC score, and then based on BIC score, if you read here, they will tell you which model is the best in your case, what current best score it generated. Then it will initialize a candidate tree set based on that selected model, and then it will generate first the candidate tree set, then it will perform some further bootstrapping to optimize your candidate tree set in this section. And then it will finalize the final tree. And okay, your final tree will be in this file. with the extension tree.file. It should be here, uh, here. This is our final tree file. This is the pairwise distance file. It should be here, this one. And this one will generate all the calculations and all those things. And this file will basically help you in your writing of thesis or papers for the methodology section. So let me download first the tree file, also the MLDS file, and also the IQ3 file. Okay. And if you are interested in a consensus tree directly, consensus tree means the program thinks this is the best tree. But remember, this tree and this tree, they will be similar. This will be the log file. Okay. Let me wait. First, we finish this step. Okay. Now we are going to analyze the distance matrix. Let me click the show code. If you can see the this file, the distance dist underscore file, it says content forward slash 0 ml dist, but our file name is different. So you have to change it, copy and only change the name. Now we are going to run it. Okay. I'm not sure you will need this analysis or not, but it actually generates the pairwise distances between these sequences. So I had 13 sequences on the x-axis, and now they put the 13 on the y-axis. And here is the distance. If the distance is smaller, it will be the dark blue. If the distance is higher, let's say 0.16 distance or the highest value, so it will be like between this sequence and between this sequence, you can see. Okay, now just a quick tip for you guys. I don't want to make this video longer, but I think this tip is important for the beginner level guys. Okay. Once you have sequences of protein sequences, so what you do, if you see if you got your sequences from the BLAST or any other database, you will see this kind of naming. So I would suggest to work on your data before aligning and before running this notebook, and fix the tree name just like I name a sequence this one albumin underscore homo sapien. So you can like albumin underscore gorilla like this. So what will happen? It will help you in two ways. If you fix the naming issue, but during fixing the naming issue, remember, do not delete this sign, the greater than sign. In bioinformatics, we call it header. If you remove this, the sequence will no longer be readable by any program, and because actually you remove the header. So just take care of this sign, don't remove it. Okay. So once your names are perfect, let's say albumin, Homo sapiens. So you just think if you use the whole data this way with the proper naming, so it will looks meaningful, and it will be easier for understanding if you're presenting this data in your research paper or in a presentation. So that was a quick suggestion. And finally, you can I'm going to open this tree file in this, how to say, bioedit i3 file. Okay, you can see here. But remember, if you want to generate this tree file in a beautiful way, so I always suggest, and I always mention in my videos, there are too many other programs, Mega and all other, but the easiest which I recommend to people you can use the iTool server and generate a beautiful tree in high resolution. So I hope you will be able from now on to generate your phylogenetic analysis and to perform your phylogenetic to generate your phylogenetic tree, and then you will also be able to write the methodology section, what you have done, how you did this. I think I fairly explain each and everything. So if you are a final request, if you are learning from my videos, I would suggest to share it with your colleagues and subscribe to my channel. Thank you so much. Bye bye.